What I am passionate about in science is that I think mainly that there's never an end to the questions that can be answered. So I do spend a lot of time in the lab, and I work with bacteria that are isolated from the soil. So I work on actually capturing those bacteria, but then also isolating individual species, and the goal is to find novel species. What I love most about my job is that there's never an end to the questions that can be answered. Even though that means that there's never really an end point, it's like there's always something to discover, and it feels really satisfying when you get a small part of the answer and you're inching closer towards understanding the world. In like working in microbiology or biomedical sciences, for me, like it feels there's this sense of pride and responsibility that I'm working towards ultimately trying to improve human life and human health, and that feels quite nice. At Bactobio, everyone is equally responsible to make things better and to like hold everyone else accountable to rigorous scientific standards. And I think everyone's opinions and suggestions are taken very seriously. So it really feels like we're part of building something together. So at Bactobio, we use mainly Starlab pipette tips and pipettes to do basically everything. Like pipette tips are ubiquitous in a lab. So preparing culture media, so that's the first step in isolating bacteria from the soil. We need to test different media types, and then from there to extract DNA and do preps for 16S sequencing of those bacterial isolates, and then for screening new bacterial isolates against pathogens to see if they produce anything that might kill the pathogens we're interested in. So really every step of the pipeline, we use Starlab pipette tips. Well, sustainability in the lab is extremely important. We do use a lot of single-use or single disposable plastic products because things like sterility in the lab are really important. But also we use very high energy equipment that need to be running all the time, freezers and incubators and things like that. And to try and find ways to reduce that is really important. So even something like instead of a -80-degree freezer, you can use a -70-degree freezer. So that sort of thing, I think, is really important. And I think there are lots of people working towards how to make labs greener, and I think that's quite important.